Hello and welcome to this webinar, Innovative Strategies for Residual DNA and Viral Titer Quantitation using dPCR. I'm Lou Rahman, Publishing Director at DDW. Leveraging digital PCR-based assays for the quantitation of residual host cell DNA or viral titers within a biotherapeutic manufacturing workflow offers value in ensuring product quality and safety. Digital PCR, or dPCR, is a highly sensitive and precise technique that enables the absolute quantification of nucleic acid targets. In the context of biotherapeutic manufacturing, dPCR provides a robust method for measuring residual host cell DNA or viral titers, which are critical parameters to assess the purity and potency of biopharmaceutical products. Also, dPCR's ability to detect low copy numbers and its resistance to PCR inhibitors makes it a great analytical tool for biopharma. So overall, leveraging digital PCR-based assays enhances the efficiency, reliability and quality of biotherapeutic manufacturing workflows, contributing to the development of safe and effective biopharmaceutical products. What you will learn from this webinar: the benefits of using the latest innovative solutions to quantitate host cell DNA or viral titers within a biotherapeutic manufacturing workflow, regulatory compliance with regards to process impurity testing, how an integrated workflow solution supports process development and a cGMP environment. The event will be presented by Jonas Bouge, Senior Manager, Product Management, and Vicky Marhan, Scientist, Molecular Biology, Thermo Fisher Scientific. A Q&A session will follow the presentations. Okay, I'll hand over to you both now.
Great. Thank you very much for the kind introduction, and thanks to everyone in the audience to join us today. Just to add on to the intro, our team of product managers, scientists, and global regulatory experts is dedicated to provide new cutting-edge solutions for the bioanalytical teams across the globe. All started over 16 years ago with our first commercial residual DNA kit. Today, we will talk a little bit about the ongoing importance of analytical testing and our broad portfolio of solutions specifically for the development of gene therapies. We will take a close look at the different PCR technologies and how to choose the right technology. I'm also very glad to have one of our R&D leaders on the call with me today. Vicky will be talking in more detail about a few recent launches and also share a bit with you how much effort actually goes into developing a great commercial kit. So let's get started. When starting your analytical assay journey, it's prudent to start with a purpose. Within biotherapeutic manufacturing, analytical assays are the cornerstone of your quality control and quality assurance. There is a lot of weight on the analytical teams to develop solutions that facilitate the verification that the biotherapeutical product meets predefined specifications and regulatory requirements, assess the consistency, purity, potency, and identity of the biotherapeutic product throughout the manufacturing process, and help ensure batch-to-batch consistency. That also help in detecting and quantifying any potential impurities or contaminants, such as host cell DNA and proteins, or residual chemicals from the manufacturing process, which could pose risks to patient safety. And lastly, the analytics should work during development, but then also meet the regulatory requirements when the program gets closer to clinic and eventually onto the market. With that, let's take a look at how we enable our customers to achieve just that. Here is a diagram of the bioproduction workflow, and along the bottom are all the products that can be used. As you see, we categorize the products for safety, quality, potency, and environmental monitoring. Among a widely adopted mycoplasma solution and our recently launched rapid sterility kit, we offer a number of viral detection and quantitation kits. We won't be talking about that today, but I recommend taking a look at them on our website. Primary focus today will be on our HEC293 dPCR kit and at our AAV titre kit that is also developed on our new digital PCR system. One very important and also very unique aspect to all these solutions is the fully integrated workflow. From sample prep over our instrumentation, consumables, all the way to our software solution. So let's start by talking about residual DNA quantitation. There are several challenges to developing and validating a residual DNA analytical assay. Developing an assay requires time, resources, and expertise, SOPs, controls, protocols, and the necessary experiments that demonstrate the assay, along with reports that support validation of the method. Validation requires compliance with the local regulatory agencies for the specific product and process. As an example here, validated per recently released ICH Q2R2. Once you have an assay, it also needs to be maintained, revalidated, etc. So in short, a lot goes into it. That's why we are dedicated to developing these type of solutions for our customers. But before we go further into the weeds, let's talk about a couple of things. First, the reason why you should be concerned with the amount of residual DNA left in your product is that we know that DNA can impact product quality, efficacy, and safety. Regulators around the globe, therefore, require you to limit the amount of residual DNA in the final dose. The World Health Organization, for instance, indicates that the amount of residual DNA per dose is kept below 10 nanograms. Residual plasmid, as an example, is also part of that 10 nanograms per dose limit, along with host cell residual DNA. For gene therapies, especially for neurodegenerative diseases, the FDA indicates that the host cell DNA content is to be kept as low as possible and that you are using a highly sensitive method to determine DNA levels. Lastly, traditional biologic productions where DNA clearance is extremely well established and also characterized, meeting these regulatory guidances and limits has not been a significant problem, but when you look at viral vector production, this has become an increasing concern, primarily due to potential encapsulation of the residual DNA into the final product. Let's stay at the topic of gene therapy, and here as an example is an AAV workflow outlining when one would conduct residual DNA testing. Residual DNA, host cell DNA, needs to be characterized. As an example, E1A is one of the oncogenes that is of regulatory concern. You should try to avoid presence of intact gene measured as a size limit of 200 base pairs. All that said, these tools are meant to enable a characterization and optimization of the manufacturing process as early as possible to mitigate and reduce long-term risks to the program and ultimately to the final product. So let's talk about risk factors and decision factors for developing these solutions in-house or by buying the expertise through a commercially available kit. It all starts with a couple of considerations. Do you have internal experts and development capabilities and expertise? An important question here is, are these teams focusing on the right thing when developing a solution that could also be bought as a turnkey ready kit? Our customers most typically want their resources and expertise go into something process and product specific. Another thing are cost considerations, which is the primary driver to start developing internally. Cost savings, one should be asking, is there a true cost savings by adding 1 to 2 FTE on an in-house developed process? How much is the true cost per test, and how much are you really saving? How much money are we really saving short, mid, and long term, and what is our trade-off between the program risks and the costs? If you develop an in-house assay, you are about 1 year ahead from implementation. You might get pushback from regulatory bodies, not necessarily on the first, but maybe on the second regulatory review on your method and coverage and not meeting the regulatory requirements. Another question to ask is, do you need customization? Right? That can reflect either process-specific or sample-specific customization that you may not want to deal with, and somebody who is developing assays for a broad market and application might have encountered those requirements and can help with either a kit that already covers it or with dedicated support. So in summary, here are the benefits of choosing a commercial kit. Commercial kits for residual DNA testing can dramatically reduce implementation timelines, reduce the need for in-house staffing, overhead, and expertise requirements. When working with us, also benefit from a global regulatory support team that works with you and directly with regulatory bodies to provide the required information about the assay and the design of it. All of this helps lower risk and results in a cost-effective solution overall. Now that you've potentially chosen a commercially available kit, let me talk about the Applied Biosystems ResDNA-seq quantitative DNA system. Mouthful here. Our ResDNA-seq DNA kits are comprehensive product solutions, including standards and reagents, ultra-high sensitivity, a highly specific, and with time to results within 5 hours. With our recent launches, we not only have a very broad qPCR-based portfolio, but also several digital PCR kits that enable the use of this newer technology. And in addition to residual DNA quantitation, we also offer lentivirus quantitation solutions. The lentiviral physical titer kit measures genome-containing lentiviral particles by RT-qPCR, and the lentivirus proviral DNA titer kit quantifies integrated proviral DNA in transduced cells for infectious titer and vector copy number assessment. Lentiviruses are relatively new to the cell and gene therapy space, so keeping up with regulatory agencies is absolute key. Also, these two assays are available now for qPCR and also digital PCR. And one of our latest products is the AAV quantitation solution. Vicky will talk about some details here in a minute. We launched this only on digital PCR and are targeting both the ITR and the SV40 region to be applicable for about 80% of commercially available AAV transfer plasmids. Before we take a closer look at the different PCR technologies, here's a portfolio of options for your residual DNA and viral titer needs. In the next section, I will talk about the qPCR and dPCR systems, the differences, and how to choose the technology for your analytical needs. Digital PCR is a method of quantifying nucleic acid targets without standard curves by dividing the bulk reaction into thousands of smaller independent reactions. PCR at large can be organized into three major categories. Bulk PCR, very traditional, our bread and butter quantitative PCRs, and now digital PCR. Bulk PCR leverages endpoint data to provide a yes-no answer. Is the target of interest present or not? Quantitative PCR provides real-time measurements and requires the use of a standard curve to provide relative quantitative data. Digital PCR does not require standard curve, and it's capable of providing absolute quantitation of known genetic targets. Because no standard curve is required, this method is considered to offer greater precision and reproducibility when compared to other quantitative methods. I always consider it as 1,000 bulk PCR reactions that you can use to absolutely quantify your targets. The key to effective digital PCR is robust, consistent compartmentalization of a bulk PCR reaction. Amplification requires all of the same major components needed for qPCR. The method used for dividing your sample into reaction volumes can have a major impact on the number, size, and consistency of the individual microreactions, all of which are key elements in data quality. After amplification, both positive and negative microreactions are simply counted. It is just binary, ultimately yielding an absolute quantitation of known targets. So now the question is, when is digital PCR a good fit? In general, for targets of very low abundance, also for monitoring small changes over time. Also when absolute quantitation is required, especially in situations where a reference standard is not easy to obtain. And lastly, when you have the need for a solution that is more resistant to inhibitors. All that said, how to choose the technology to use for residual DNA and titer assays. dPCR is still somewhat emerging, where qPCR is very well adopted, also in the minds of regulatory agencies. qPCR offers a very broad dynamic range, whereas digital PCR shines with high sensitivity and no need for standard curves. For residual DNA specifically, qPCR offers a readout in mass, whereas digital PCR readouts require a conversion from copy number to mass. We touched base on the standard curve requirements already, as well as on the regulatory perspective. qPCR is mostly used with sample prep, including a DNA extraction step. Digital PCR often doesn't require the extraction step and dilutions of the sample to dilute inhibitors and move the concentration into the dynamic range is often sufficient.
Hello everyone, my name is Vicky. I'm a scientist at Thermo Fisher Scientific. Today I will introduce to you two kits that we have, the HEK293 DNA kit and the AAV kit, and I will show you some data that has been generated using the two kits. Assay in our portfolio, which is the dPCR HEK293 DNA kit. There are some challenges when designing the assay for HEK293, and below we have outlined some of the key considerations for us when we design this assay. First is the sensitivity requirements, where we have to focus on high copy target gene to detect and quantify trace amounts of HEK293 DNA. Secondly, we need to ensure that there is wide chromosomal coverage for HEK, so we have to use target genes that are distributed across the chromosomes to ensure broad coverage. Thirdly, we need to identify targets which have less variability for accurate quantitation, so as to ensure that there's distinct populations between the positive and the negative population in the digital PCR plots. Lastly, for the non-template controls, we also have to ensure that there's minimal background in the blank samples with a limit of detection that is well above the limit of blank. The assay can be used to analyze HEK293 host cell DNA quantities to adhere to the regulatory guidelines. The performance specifications of the assay, as outlined here, is well suited for this purpose because it has a broad dynamic range, which is from 18 to 18,000 copies per reaction, which is about 0.7 to 7,000 picogram. And this is supported by good linearity, speaking to the accuracy of the assay. And importantly, the kit is highly sensitive with the LOQ at 1 picogram per reaction and a LOD at 0.7 picogram per reaction. And these specifications ensure that there is excellent separation throughout the dynamic range as indicated in the plot here. The analytical validation was conducted using the HEK293 genomic DNA. The HEK targets are read on the FAM channel, whereas the internal positive control is read on the VIC channel. The results across different quantitation points, as indicated in this column here and in this column here, showed high accuracy as well as good precision through the different quantitation points, and this is supported by distinct separation seen in the plots between the positive as well as the negative population. The internal positive control demonstrated consistent readout across the different dynamic range, making it a reliable indicator for inhibition, in particular in complex matrices with high salt or protein concentration. To minimize the background noise in the blank sample and to have a clear separation between the limit of detection and the limit of blank is important for accurate assay performance. In the table here, we can see that the average limit of blank across the 68 wells was 0.19 and the average LOD across the wells were 2 copies and there were 100% detection with all the points well above the limit of blank. The plots that are shown below also indicate a clear separation between the LOB and the LOD with no overlap in signal. This separation confirms the assay's ability to reliably distinguish between the true positive signals and the background noise. Given that the HEK293 cell line used in this assay is of human origin, it is important to conduct the experiment in a clean environment to avoid any potential contamination. This will include following appropriate laboratory practice and to wear suitable PPE to also ensure the integrity of the results that are obtained. In cell and gene therapy, accurately detecting AAV genomes is vital for ensuring the efficacy and safety of treatments. Our duplex assay detects both ITR2 and SV40 genes within the AAV genome and can be used to assess the virus titer in sample. The assay is a comprehensive solution comprising of all the necessary digital PCR reagents that are required. It is accurate, sensitive, as well as highly specific, and no cross-reactivity is observed for host cells and non-AAV vectors and other impurities. Lastly, the two targets, which is ITR2 and SV40, they are highly conserved genomic regions, with the design being compatible with over 700 commercial transfer plasmids. The ITR2 assay is read on the FAM channel, whereas the SV40 assay is read on the VIC channel. In addition, we also have the internal positive control, which can be read on the AB channel. The assay specifications for the AAV DNA titer case is outlined here. Good linearity is observed across different quantitation points, and there's high precision as well, where the CV percentage across different points is less than or equal to 20%. The assay is highly sensitive with a limit of detection at 9 copies for ITR2 and SV40 and the limit of quantitation at 27 copies for both targets. The assay has a broad range that is between 9 to 90,000 copies. The ITR2 is read on the FAM channel and the SV40 is read on the VIC channel. And throughout the different quantitation points, excellent separation is observed, ensuring that the quantitation is accurate and robust. The assay also has an internal positive control that is read on the AB channel, which is a good indicator for assay inhibition. The validation of the AAV assay involved the use of crude ATCC reference standard material AAV8 without DNase treatment and extracted using the Kingfisher Flex purification system. The results of the assay demonstrated high accuracy and high precision across various quantitation points for both targets, ITR2 and SV40, as indicated in this table. This finding suggests that the assay exhibits high reproducibility when quantifying high viral samples, ensuring reliable and consistent results across multiple runs. Additionally, the internal positive control consistently provided readouts, making it a valuable tool to indicate any inhibition in the runs, in particular when working with samples containing complex matrices. By including an internal positive control, the assay can effectively identify any potential issues that can affect the accuracy of the results. Assessed for robust sensitivity by spiking in 1 copy in 64 wells. All wells tested showed 100% detection with no fallouts. This indicates that the assay is highly sensitive and capable of reliably detecting low levels of targets. In addition, the non-template wells were included in the testing to determine the limit of blank. It was observed that no amplification occurred in these wells, indicating that there was no false positive signal or background noise that could interfere with the accurate detection of the target. The plots indicated here show a distinct separation between the positive and the negative population at low copy numbers.
Thank you so very much, Vicky. And before we jump into our Q&A session, I want to give you a few things on your hand when you're making your decision for your analytical solution. So first, with over 1,400 service and support specialists worldwide, we support you in your journey wherever you're going. We offer validation and implementation guidance, as well as bioinformatics and IT services to make sure things are going to work according to your plan. We have our team to come out and train your teams to meet both regulatory guidance and also to become expert in those fields. When you're approaching your regulatory agencies, we support with regulatory expertise and support, often working on behalf of our clients and customers directly with regulatory bodies to provide the required information. And then we offer service plans for our instruments to make sure things will work this year, next year, and going forward. With that, I'll hand it over, and we are looking into a couple of questions that the audience has submitted. Thank you.
Thank you both for that. That was a great webinar. And before we go over to questions with Jonas, I'd just like to remind everyone that the webinar will be available to watch on demand at www.ddw-online.com. If you're ready, Jonas, we have some questions for you.
Yes, thank you, Lou.
Great, okay. So the first one that we have is, can the same results be expected for the same assay on WOVE and dPCR?
So if we have the same assay, and you are using a qPCR and a digital PCR instrument, then I would say the same results can be mostly expected with a little asterisk here. You need to quantitate your control in the same way, right? If you use different approaches to quantitate your control and then use your control both for your relative determination on qPCR and for digital PCR, you may end up with different results. So therefore, I always recommend when you're using a control, quantitate the control on the digital PCR side.
Okay, lovely. Thank you for that. And the next question we have is, what are the gene targets of your residual DNA? Is this only for HEK293? And what about minimising resistance or ORI?
I think there are probably two questions. Let me answer the first one, right? The HEK293 kit is, well, I can't specifically share what that we are targeting in our assays, but I can share that it's a highly repetitive element, multiple highly repetitive elements that are across all the chromosomes for multiple reasons, right? Primarily to achieve the sensitivity that we are achieving. Secondly, it's a targeting chromosomes across the entire human genome, which makes it specific to human cell lines, not only specific to HEK293. If we would only go after something that is specific to HEK293, would not get to any sensitivity that is really required. So that's why assay is specific to human and not just to HEK. And the second part of that question, kanamycin resistance or ORI, we don't have this as a digital PCR solution just yet, right? We have a kanamycin resistant plasmid quantitation solution as part of our qPCR portfolio, which we do see adopted very often. It's certainly something that on the digital PCR side is certainly something that it's valuable to add to our portfolio at some point. We don't have it just yet.
Okay, lovely. Thank you for that answer. The next question is sample prep extraction required?
I would say yes and no. I think Vicky tried to speak also a little bit to that, right? And I would say in most cases, and most customers that I see in the field don't need sample extraction. Dilutions are sufficient enough in most cases. There are samples that are containing so many inhibitors with a low abundance target that diluting enough is not possible. And in those cases, extracting the DNA is the better course of action, right? If you really have a low abundance target with a lot of inhibitors. That's typically what we're seeing as being the samples that require extraction. But as I said, in the vast majority of samples, and I think that goes across all different technologies on the digital PCR side, extraction is typically not necessary.
Okay, thank you. And the next question is the HEK assay a single or multiplex assay?
It is a multiplex assay, right? As I said in the previous one, multiple targets, all of them are repetitive elements across, so multiplex to increase the sensitivity.
Okay. And what's the difference between the qPCR HEK kit and the dPCR HEK kit?
So our qPCR kit, extremely well adopted, is targeting a target that is even more abundantly present across the entire human genome. And it was so abundantly present that this, as an assay, would not have worked on digital PCR because you would have just gotten too many positive individual wells. That's why we redesigned the assay specifically for digital PCR to reduce the number of replicate targets, but still have a very high number of replicates to ensure that we can meet the sensitivity. So different targets is the short answer, but both are repetitive and highly representative.
Okay, thanks, Jonas. And then there's one final question that's just come in. It's which AAV serotypes can you analyze with the AAV kit?
The AAV kit is serotype agnostic because we don't have the standard in the kit, and it's really going after targets that are present in, we measured around 80% of commercially available plasmids with targeting ITR and SV40, so serotype agnostic.
Okay, thank you. Thank you for answering those questions from the audience, and thanks again for talking to DDW today. I'd just like to remind everyone that you can listen to this webinar again on demand at www.ddw-online.com, and you can now view Drug Discovery World content online or on your mobile or tablet. Thank you for listening.
Thank you.